University in 2004. He was the math champion of Bangladesh. And I have to emphasize that's the with capital T. I don't know what happened in 2002. Maybe he could say he took an off year. And then after that, he got a degree with electrical engineering from the Bangladesh University of Engineering and Technology, and then with various stints in the UK and then US, eventually came here and joined my lab, and he's been just doing amazing things. I think Shoaib has been a great contributor to all aspects of the lab, besides the intellectual environment. I think just from a cultural side, he's really contributed to the cultural fabric. We've learned a lot about Bangladesh culture. I think it's also been funny because we've been introducing him to US culture, and that gives us sort of a chance to introspect a little bit. Like, actually, I'm not going to talk about the gold digger story. I think that, we'll save that. He can talk about that later. And I think in the end, I would say that, Shoaib is one of the most giving people that I know. I know that even outside the lab mates, with people in the Bangladesh community, with people in the community of his mosque, some of whom are here, I know that he is very generous with his time. He's been extremely patient and a great teacher. And this has been especially helpful working with someone like me. And I have to say, there's been a lot of times where I've said to him, Shoaib, let's pretend I don't understand your equations. How can we talk about this in some other way? And with that, I think it's going to be clear the intellectual rigor and thoughtfulness that Shoaib has brought to his work. And I welcome Shoaib to talk about all the great work that he's done.
Thank you so much, Jeff. It was a great introduction. And thanks everyone for coming. So today I will tell you about how we integrate sensory information over our hands. So we study touch in our lab in general, and we are interested with touch because it allows us to interact with the external world. For example, object manipulation, texture perception, and social interactions. And we do these things almost always in our daily life. So when we interact with this world, our skin receives a lot of information, and that information is transduced into our skin through these mechanoreceptors. And one of the important information that our skin receives is the vibration frequency. And this is specifically important for perception of textures. So for example, different textures generate different patterns of vibrations on our skin, as indicated by these spectrograms of two different textures. And these high frequency vibrations are transduced into our skin through these Pacinian mechanoreceptors. So now, what happens if we do not have the sensation for touch? So here is a demo. So in this video, in the left side, the person is trying to light the matchstick, and she can do that very easily, you can see. Now, in the video, on the right side, the same person is trying to do the same thing, but this time, her hand is anesthetized locally, so she cannot feel the cutaneous feedback. Now you can see that she is really struggling, picking up the stick, holding it, and keeping the balance, putting it on the matchstick. She's trying really hard, and finally she makes it. So this type of terrible things may happen if we do not have the sensation for touch. Now, we can touch something without one hand or two hands. But if you look for the literature, then you will find a lot of studies that have been focused to understand the touch with one hand or unimanual touch. But a little is known about touch with two hands or bimanual touch. So that means, yes, we are so advanced with the one hand, but we are not so rich with two hands. So now the question is, why do we care about two hands? So let's just start with an example. So this video demonstrates an amazing use of our two hands. And in order to do this task, our nervous system requires to coordinate actions between the hands and at the same time integrate sensory information over the hands. And this is amazing to me. And I do not know how many of you can solve a Rubik's Cube like this, but we can, almost all of us can do some more common activities like buttering a shirt or tying shoelaces. And all these tasks require an extraordinary amount of coordination between the two hands. However, although a lot of attention has been dedicated to understand how we control and coordinate actions between the hands, a little is known about how we integrate sensory information over the hands. So my thesis has been dedicated to understand this bimanual touch. Specifically, I used psychophysics, modeling, and computational neuroimaging to understand how we integrate sensory information over the two hands. So today, I will tell you some of the most exciting results that I found in my last 5 years. So in the first part of my talk, I will show you how we perceive vibration frequency information from two hands. And I will specifically address 3 questions. So the first question is, how does the perception of vibration on one hand depends on what we experience on the other hand? Second, do bimanual interactions depend on the relative positions of the hands? And this is important because we can move our hands in the space. So that means the same receptors can be activated even if the positions of the hand varies in the space. And the third question is, what are the computations that mediate these interactions between the two hands? So we address these 3 questions using behavioral experiments and modeling. So the fundamental units of our behavioral experiment is a frequency discrimination trial. On a given trial, participant given 2 vibrations on their right thumb. It's here, like zzz, zzz. So the task was to report which of these 2 vibrations was higher in frequency. In this case, the first one. Now, in the actual experiment, one vibration always contained a standard 200 Hz, and the other stimulus contained that is a compression stimulus whose frequency varied between 100 and 300 Hz. And this is the target stimulus presented on the right thumb. Then we presented a distractor stimulus on the left index finger, but the participant was instructed to ignore whatever happens on the left hand. Now, by varying the distractor frequency, we can understand how a standard 200 Hz is perceived when paired with 100 Hz, 200 Hz, and 300 Hz. And the other stimulus was congruent with the compression. Now, in addition to that, we also manipulated the positions of the hands from far to middle to near to understand position-specific interactions between the hands. So then we characterize the performance of this task using choice probabilities, which is defined as the probabilities of judging compression frequency higher than the standard frequency. And in this plot, you can see that when the compression frequency is 100 Hz, then participant never said that that is higher than the standard 200 Hz. In contrast, when the compression frequency is 300 Hz, then participant always says that this is higher than the standard 200 Hz. And the probabilities are something between 0 and 1 for the intermediate frequencies. And then we fit these probabilities using psychometric function and characterize the behavioral performance using 2 parameters from this psychometric function. So the first parameter is the point of subjective equality, or PSE, which is the compression frequency that is perceived equal to the standard frequency. This is a measure of bias. The second parameter is the JND, or just noticeable difference, and this is the amount of change with respect to this PSE to make a noticeable difference. And this is a measure of perceptual threshold. A higher threshold means bad performance. Okay, so now here I'm showing the performance of a single subject at the far position of the hand at the baseline condition, meaning that when there was no distractor stimulus presented on the left index finger. And in the presence of distractor, we can see that the curves clearly shift to the left or to the right depending on the distractor frequencies. And this is for the far position of the hand. And this pattern is consistent for the other 2 positions of the hand, except that the effect size increases as the hands get closer. Specifically at the near position here, we can see a bigger shift to the left or to the right of the curves depending on the distractor frequencies. This is for a single subject. And we tested 8 participants, and all of them showed this same pattern. And those are indicated by these thin lines, and the thick lines indicate the group average. Now here I'm showing the quantification of this behavioral performance. So this is the change in bias of TSE, and this plot shows how the distractor on the left hand bias the perceived frequency on the right hand. And we see that the bias increases, or the bias becomes larger for 300 Hz and also for 100 Hz as the hands get closer. And it becomes maximum at the near position, which means that at the near position, if we present a 200 Hz vibration on the right hand and 100 Hz vibration on the left hand, then the perceived frequency on the right hand will be approximately 150 Hz. But if the two frequencies on the two hands are identical, then we do not see any change in bias. So then here I'm showing the change in threshold of JND, and once again this is a measure of how bad the performance is in the presence of distractor. And the positive values in this plot means the distractor in general impair the performance. And once again you see that threshold increases for this 100 and 300 Hz as the hands get closer. But interestingly, when the distractor is 200 Hz, then we see opposite pattern. Specifically, it just decreases as the hands get closer. And specifically in the near position, we see that the threshold does not change. Whereas the threshold is maximum at the near position for other two conditions. So these two results together imply a complex interaction pattern between the distractor frequency and the positions of the hand. So now we want to understand this interaction pattern. And in this case, we need a little help from math. But the idea is very simple. So if we have the target stimulus that is represented using this type of distribution, this is basically Gaussian distribution, and also the distribution for the distractor stimulus, then these two distributions will be combined to generate the final percept with some computations. And here is the model that we use to infer these computations. So once again, this is the target stimulus, this is the distractor stimulus, and these are the distributions. Now the combination of these two distributions depend or determined by 3 components in this model. So one component is the frequency-based modulation function that changes the reliability of the distractor and also the target. And this can be considered as the feature-specific modulation. Now the other 2 components determine the weights to the target hand and to the distractor hand. In the first step, a location-based weighting function determines the weight to each hand independently. As we can see that in this plot, when the hand is the near position indicated by 0 here, both hands are weighted equally. But if when the distance increases, then the weights to the distractor hand reduces. So this is in the first step. Then in the second step, these weights are then updated as the weights relative to each hand by a technique called normalization, which is also called divisive normalization. And this type of canonical computation has been reported in the visual systems and also in olfactory systems. Now, these 3 components combine these 2 distributions to generate the final percept, which is theta here. And this is basically the target stimulus shifted towards the distractor. And this model has only 4 parameters, and we will use this model to explain the variance of 63 data points. So if we take this model using a cross-validation approach, which means that the full data set was divided into 2 halves. First half was used to train the model, and the prediction was used to test with the second half. Similarly, the prediction from the second half was used to test with the first half. Now, we can see that this model can explain more than 90% of the variance in the data for within subject cross validation for each subject, and the average is 94%. Now we had 8 participants. Now if we train the model with the data from 7 participants and test the model with the remaining participant, with that across subject cross validation, we can see that the variance explained is 96%, which is very high for this type of data. And in fact, this is close to the noise ceiling, meaning that no other model will perform better than this model given our data. And in fact, we tested a total of 19 models, and we found that this model was the most probable. Now, with that, I would like to summarize what I have shown you in the first part of my talk. We have seen that the perception of vibration frequency on the right hand depends on what we experience on the left hand. Then we have seen that the interactions between the hands depend on the relative positions of the hand in the space. And finally, we have seen that the interactions between the hands can be understood by a Q combination model. So this is all about frequency perception. We also found that this type of interactions happen for intensity perception, but in a different way that we have seen for the frequency perception. So that means we use some sort of feature-specific computation that depends on whether we are perceiving frequency or we are perceiving intensity. But that's a separate story, and for the interest of time, I'm not going to details of this result, but if you are interested, then I'll be very happy to talk in the end. So from this first part of my talk, now we know how we perceive vibration frequency from our 2 hands. Now the question is, where and how does our brain represent these vibration frequency information? So that I'll be talking about for the rest of my talk. So in our lab, we use functional MRI to measure the brain activity. So this is the image of a brain, and for the better visualization, we will use this inflated brain. Now, to understand the frequency representation in this brain, we performed experiments in which, in a given scan, participants receive vibration either on their left hand or on the right hand, or both and simultaneously. And this process continued for the rest, for the whole scan. And we used 9 different vibration frequencies for the left hand, 9 different vibration frequencies for the right hand, and a combination of 15 frequencies for the both and simultaneously. Now, during the experiments, participants were instructed to attend only to the right hand, similar to our psychophysical experiments. And during the experiment, participants also performed some sort of frequency discrimination task just to make sure that they were involved in the experiment rather than, let's say, sleeping in the scanner. Okay, so this type of experimental design allow us to characterize both unimanual and bimanual responses in the brain. That we will see one by one, but first look at the data. So here I'm showing the activity pattern in the brain based on this vibration stimulus. So the responses of this colored area was modulated, or the responses of this colored area were modulated by the vibration frequencies presented on the hands. And we can see that there are a lot of activities in the lateral parietal cortex here and in the frontal cortex in the both hemispheres. And we do not see much activity in the primary somatosensory cortex. But this pattern is consistent with what we found in other experiments in our lab and also in other labs. And more importantly, this pattern is consistent for all the participants in this experiment. So now the question is, how the responses of these areas relate to which hand was stimulated and what frequency was used to stimulate the hand. So in the first step, I will show you how these responses relate to which hand was stimulated. And to do that, I'm showing here the response, the bold responses for a single box cell, which is the population of neurons for 9 different vibration frequencies for the left hand, 9 different vibration for the right hand, and 15 conditions for the both hands. Then we took the average of the absolute responses, ignoring the direction of these responses, separately for the left hand, for the right hand, and for the both hands, to compute something called modulation index. Now, this allows us to characterize each box cell just by these 3 values. And we did that for all the box cells in these activated areas and represented in this heat map, where each row represent a box cell. And then we clustered and sorted these box cells and found that there are 5 distinct classes of box cells in these activated areas of the brain. So one of the classes respond only to the left-hand stimulation. One of them responds only to the right-hand stimulation. And one of them responds only to the both-hand stimulation. Then there is another class that respond only either to the left-hand stimulation or to the right-hand stimulation, but they do not respond when both hands are stimulated simultaneously. And the final class that respond to right-hand stimulation or both-hand stimulation, but no response to the left-hand stimulation. So this finding is already a striking finding, and importantly, these 5 classes of box cells are consistent across these 5 participants. And we also found that these 5 classes of box cells are distributed randomly in these activated areas of the brain. So it looks like a salt and pepper type of distribution rather than a cluster distribution.
Were they all right-handed?
So that's a good question. Yeah, so all the participants are right-handed, yes. Okay, maybe we can discuss more on that right-handedness things later. Okay, so yeah. So this distribution looks like a salt and pepper type of distribution rather than a cluster distribution, which means that both hands are represented almost everywhere in these activated areas of the both hemispheres. Okay, so now in this analysis we just considered which hand was stimulated, but we ignored the frequency information. Now in the next step we want to understand how these response patterns relate to which vibration frequency was used to stimulate the hand. So the idea here is, if we have some vibrations presented on the hands, then by some operations we will see this type of we'll observe this type of response pattern for a given box cell. So the way that we try to understand these operations is by using the encoding model. And once again, this encoding model will involve some mathematical equations. So here I'm showing a very high-level description of the encoding model. So this is the predicted responses, and the model has 3 components: left hand, right hand, and both hand components. And for these unimanual components, we assume that each box cell contains neural populations or channels that are tuned to vibration frequencies. And these each channel contribute to bold responses for the left-hand stimulation depending on their weights. So a separate set of channels and weights contribute to the bold responses for the right-hand stimulation. Now, for the both-hand stimulation, we used bimanual pin integration technique, which means that if we have 2 vibrations presented on the 2 hands simultaneously, let's say a lower frequency presented on the left hand and a higher frequency presented on the right hand, then based on the channel responses, we create some sort of likelihood distribution, one for the left hand and another for the right hand. Then these 2 distributions are combined linearly to generate the likelihood distribution for the both hand stimulation. And this distribution will contribute to the board responses for the both hand stimulation, depending on different weights. So we fitted this full model, again using the cross-validation technique that I have shown you in the first part of my talk. And here is the result that we found. We fitted this model for each voxel in this entire brain. And we found that the responses of these red areas were significantly explained by this encoding model. And once again, we see that these red areas are mostly located in the lateral parietal cortex and also in the frontal areas. So which means that there's some sort of consistency between this encoding model analysis and the traditional analysis that I have shown you a couple of slides ago. And importantly, once again, this pattern is consistent with all of our subjects. And we also confirmed that the goodness of fit of this model, which is the correlation coefficient, was close to the noise ceiling, which means that no other model will perform significantly better than this model given our data. So now we have an encoding model that is working. Now the question is, what can we learn from this encoding model? So I will tell you two things that we can learn. The first thing is we can understand how we can understand unimanual vibration frequency representation in the brain. And second, we can understand the bimanual computations. So in the first step, I will show you how we can use this encoding model to understand unimanual representation in the brain. So to do that, we computed the frequency response profile for all these voxels in the red areas. And here I am showing that frequency response profile for all the voxels for a single subject for a single hand, where each voxel, where each row represents a voxel, and the color of this plot represents the response modulation for these different vibration frequencies. And the voxels are sorted according to their best modulating frequency. And we can see that the voxels are tuned to vibration frequencies that covers the entire range of frequencies that we tested in the experiment. So on the right side here, I'm showing some of the example voxels that are tuned to lower frequencies, intermediate frequencies, and higher frequencies. And the black dots in this, black dots here represent the observed board response, and the blue lines indicate the estimation from our encoding model, and the red lines indicate the Gabor fit. And it is interesting that we can now fit these responses using Gabor function, because now we can characterize this type of responses where we see sharpening frequency of where peak and to Planck inhibition. And this type of responses have been reported in the visual systems and also in auditory systems, and we have seen that this type of thing to be characterized by Gabor function. So this is, for the very first time, we see any evidence of vibration frequency tuning in human somatosensory cortex. Nobody has shown vibration frequency tuning, not even in non-human primate or any somatosensory cortex, except that very recently, or earlier this year, a group reported similar type of frequency tuning in mouse somatosensory cortex. Now, given that we can characterize this type of responses using the Gabor function, now we can analyze the parameters of the Gabor function to understand the frequency representation in the brain. So I'll give you just one example how we can use these Gabor parameters, and that parameter is related to the best modulating frequencies. So we computed the best modulating frequencies for all the significant voxels, and here I'm showing the distribution of that voxels for each subject, which are indicated by the colored curves. And the black curve indicates the group average. And this is for the right hand stimulation, this is for the left hand stimulation. And here we see that many of the voxels are tuned to a frequency nearly 220 Hz in both hands. So this is important because our afferent systems are most sensitive to a frequency of approximately 250 Hertz, which means that our brain is optimized to process the information that is most available to the brain from the periphery. We also wanted to know if there is any topographic organization of this modulating frequencies or not. And here is the result that we found for all of our five participants, and we believe that there is no evidence for topographic organization that is systematic across these participants. Specifically, if we compare that with the tonotopic organization in the auditory cortex, where we see high frequency, low frequency, and high frequency representation, it would be awesome if we would get that type of topographic organization in the somatosensory cortex, but we don't. So this is just one example that we can use the Gabor function to talk something for the very first time about the frequency representation in the brain. We also analyzed some other parameters, those are related to tuning asymmetry, frequency selectivity, and response modulation. And we found that the results are consistent across the participants. And the results may also reflect some special attention effect, but I'm not going into details of that. But please feel free to ask any question if you're interested in the end. So this is the unimanual representation, just by using the encoding model. So then I'll show you how we can use the encoding model to understand bimanual computations. So this is the encoding model that I showed you a couple of slides ago. And this time I will mostly focus on this part, the bimanual Q integration. And just as a reminder that for this bimanual integration, we use some sort of integration of these likelihood distributions. And those are complex operations. Now the question is, do we really need these complex operations? Or can we simply use some more simple operations like conjunctive coding, in which the bimanual responses will be predicted simply based on the unimanual left response and unimanual right responses? We have been seeing this type of conjunctive coding in visual systems and also in auditory systems, but we know the conjunctive coding of different features. So one other thing about this model in this case is that the unimanual part is identical to the Q integration model. So the only difference between the Q integration model and this conjunction model is how the bimanual responses are predicted. So we figured this model again with using cross-validation approach, and here is the result that we found. So this is the performance comparison between the two models. In this plot, I'm showing the percentage of box cells explained by each encoding model. And we see that a vast majority of the box cells are explained by the integration model only. And a tiny amount of box cells are explained by the conjunction model only. And a significantly fair amount of box cells are explained by both models. Now, by definition, these two models are identical for the unimanual responses. And we confirmed that from a separate analysis. So the differences that we are seeing between these two models must come from the bimanual parts. And here I'm showing the proportion of box cells explained by each model for different classes of box cells. Just as a reminder, we identified these 5 classes of box cells in our previous analysis that are represented in this x-axis. So we see that the conjunction model, which is represented by this blue card here, is really failing to explain the B class box cells and RB class box cells. And these are the bimanual classes. And we also found that the prediction error is higher when both hands are stimulated, specifically for B class box cells and RB class box cells for conjunctive coding model. as compared to the integration model. So these two analyses show that the bimanual responses are more consistent with the Q integration rather than the conjunctive coding model. So now I have shown you that how we can use the encoding model to understand bimanual computations. So finally, I will show you one more analysis that is more application oriented. So we have an encoding model and that is working. So now the question is, can we use this encoding model to decode our brain? So the idea here is we have some activity pattern in the brain. Can we use that to understand what vibration frequency was presented to stimulate which hand? So here I'm showing the results of the encoding. So this is the true event, this is the decoded event, and the high values in the diagonal represents the higher accuracy. And we see that all of the participants showed a higher accuracy in general. And here is the quantification. We say that the overall decoding accuracy is approximately 55%, which is way better than the chance level, which is just 3%. And we also showed that the unimanual conditions and bimanual conditions were separately decodable, which means that this overall accuracy is not dominated by any specific conditions. And finally, we also showed that the decoding accuracy increases as the number of boxes included in the decoder. And we see that with approximately 40% of the boxes, we can achieve an accuracy that is higher than the chance level. And importantly, these percentage of boxes were selected randomly from different locations of the brain. So that again suggests that the information related to the unimanual responses and bimanual responses are distributed almost everywhere in the activated areas of the brain. So with that, I would like to summarize what I have shown you today. We have seen that the perception of vibration frequencies on one hand depends on the vibration frequencies that we experienced on the other hand. We have also seen that the strength of the interactions between the two hands depend on their relative positions in the space. And in the second part, in the neuroimaging studies, we have seen that both hands are represented almost everywhere in the activated areas of the brain. And we have also seen that boxes are tuned to vibration frequencies, and that is the very first time we see any evidence of vibration frequency tuning in human somatosensory cortex. And finally, we have seen that the bimanual responses are more consistent with Q integration technique rather than the conjunctive coding technique. So now the final thing is, why is this not important? So certainly we can use these findings to advance our textbook knowledge. But more important thing, at least to me, is that we can use our findings to develop some more real-life applications. So for example, in this video here, I'm showing that a robotic arm is picking up an object and placing that in a separate location. And the fact here is this arm does not receive any